Hello everyone, I would like to introduce myself. My name is Mike Cristero Sobreno from University of Don Mariano Marcos Memorial State University, Mid La Union Campus. So we will discuss about the biotechnology.
Biotechnology. Biotechnology is the manipulation of biological organisms to make products that benefit human beings. It contributes to such diverse areas as food production, waste disposal, mining, and medicine. Modern achievements of biotechnology include the transfer of a specific gene from one organism to another by means of transgenics, the maintenance and growth of genetically uniform plant and animal cell cultures called clones, and fusing of different types of cells to produce beneficial medical products such as monoclonal antibiotics, which are designed to attack a specific type of foreign substance. The first achievements in biotechnology were in food production. Occurring about 5000 BC, diverse strains of plants or animals were hybridized to produce greater genetic variety. The offspring from these crosses were then selectively bred to produce the greatest number of desirable strains. Repeated cycles of selective breeding produce many present-day food production programs. In the 1960s, an important project used hybridization followed by selective breeding to increase food production of quality of wheat and rice crops. Biotechnology is science of using living organisms to improve human life, and it is something we experience every day. From the food we eat to medicines we take, biotechnology plays a big role. Long ago, farmers practiced early biotechnology by crossing plants and animals to create better varieties, leading to stronger crops and healthier livestock. Today, scientists have advanced these ideas through gene transfer, cloning, and producing medicines like monoclonal antibodies to fight disease. Even the Green Revolution in the 1960s, which improved rice and wheat, shows how biotechnology helps fight hunger and create a better future. The term of biotechnology was first used by Karoli Ariki in 1919 to refer the production of products from raw materials with the aid of living organisms.
Hi, my name is Jerome B. Picardal. Today, I'm gonna discuss the 5 animals cloning. Animal cloning. Animal cloning is the process of creating a genetically identical copy of an animal using somatic cell nuclear transfer SCNT or other techniques. Purpose: reproductive biology research, understanding development and reproduction. Conservation, preserving endangered species. Agriculture, improving livestock breeding in genetics. Veterinary medicine, developing new treatments and therapies. Techniques, somatic cell nuclear transfer SCNT, transferring the nucleus from an adult cell into an egg cell. Applications, livestock improvement in chasing desirable traits. Endangered species, preservation increasing population numbers. Biomedical research, developing new treatments and therapies. Dolly the sheep was the first mammal cloned from an adult cell using somatic cell nuclear transfer SCNT. She was born on July 5, 1996 at the Roslin Institute in Scotland. Dolly's cloning marked a significant breakthrough in genetic engineering in reproductive biology. Comolina the mouse was the first mouse cloned from an adult cell using somatic cell nuclear transfer in CNT. She was cloned in 1998 by a team of researchers at the University of Hawaii. Molina's cloning demonstrated the feasibility of cloning mammals using adult cells and paved the way for further research in the field. Snuffy the dog was the first dog cloned from an adult cell using somatic cell nuclear transfer in CNT. He was cloned half-grown hound born on April 24, 2005 at Seoul National University in South Korea. Snuffy's cloning marked a significant achievements in canine cloning and opened up new possibilities for research and application in veterinary medicine and conservation. Snow Wolf the wolf was a cloned gray wolf. An endangered species, Snow Wolf was cloned using somatic cell nuclear transfer in CNT in 2007 by a team of researchers at Seoul National University in South Korea. These cloning achievements demonstrated the potential for cloning to contribute to conservation efforts for endangered species. Promethea the horse was cloned half-Langer horse born on May 28, 2003. At a laboratory in Italy, she was the first horse to be cloned using somatic cell nuclear transfer in CNT. Prometheus cloning marked a significant achievements in equine cloning and opened up new possibilities for research and application in veterinary medicine and horse breeding.
My name is DJI of Francia and I live in from Orbistondo, San Juan, La Union, and I'm 21 years old BSIT student. I'm gonna discuss the 5 animal cloning. CC the cat, 2001. The first cloned cat in the world. Made in the United States. Her name means copycat. She was cloned from another female cat using somatic cell nuclear transfer. Idaho Gem the Mule, 2003. The first cloned mule and the first clone of a hybrid animal. Clone at the University of Idaho from a champion racing mule. Sam the Goat, 2009. A clone goat created to produce valuable proteins in its milk, used for medical and scientific research. Garima the Buffalo, 2010. The first cloned buffalo in India, created to improve milk produce and breeding programs. Peng Peng the pig, 2012. The first cloned transgenic pig in China, created for biomedical research and to study human diseases.
My name is Marian Kalika and I'm gonna discuss the genetically modified organism GMO crops. These are plants that have been scientifically altered at the genetic level their DNA. Using modern biotechnology, scientists insert, delete, or modify specific genes in these plants to give the new traits. This process is done to improve the crop's quality, productivity, and resistance to challenge in farming.
Good day to all of you. I'm Carl Aaron Likod, and today we will talk about GMO crop.
First, corn. Corn was genetically modified to resist pests such as corn borers and to tolerate herbicide like glyphosate. This allows farmers to spray their field to kill weeds without harming the crop, which reduces the crop loss and increases yield. As a result, it becomes cheaper and easier to grow corn on massive scale, making it highly profitable for large farming operations and food industries that rely on corn animal feed, biofuel, processed food product. Second, soybeans. Soybeans were genetically modified primarily to tolerate herbicides, enabling farmers to control weeds more effectively without damaging the crops. This led to a higher productivity, reduces labor, and more efficient farming practices. Since soybeans were major source of animal feeds, cooking oil, and processed food ingredient, GMO technology helped meet the high global demand and support large-scale agricultural production. Third,
Apples. Apples were genetically engineered to prevent browning when sliced, which improves their appearance and extends their shelf life. This makes them more appealing to consumers and reduces the amount of fruit wasted by grocery stores, restaurants, and households. Fourth, papayas. Papayas were genetically modified to resist the ring spot virus, a disease that once threatened to destroy the papaya industry, especially in Hawaii. Development of virus-resistant papaya not only saved the crop from being wiped out, but also ensured the survival of papaya farming and the livelihood of many farmers. Lastly, rice, especially the golden rice, was genetically engineered to produce beta-carotene, a source of vitamin A to help fight a vitamin A deficiency countries where rice is a staple food. This biofortified rice was the potential to improve public health and reduce malnutrition-related diseases. At the same time, it allows biotech companies to maintain control over a vital global food source, showing how GMO technology can serve both humanitarian and economic purposes.
Sayang, kamu juga sayang.